Okay, folks, let's get started. It's nice. It's kind of courtesy when people ahead of you clean up their messes. When they don't clean up their messes, that's not good. Why do we only have one screen? Don't know. We also have a bouncing ball, don't we? All right, so how's your exam preparation coming along?
Great.
Great. That's better than I heard yesterday. Talk to me. What you thinking? You want more material in the next exam or what? No? The usual. Okay. All right. So I haven't written the exam yet, so what should I put on it? One question. You want one question? Seriously? You can kill him. And I'm guessing you want to give me the question, too. Yeah. So like I said, look at the format of the exam I've posted. The format of your exam will be exactly that. And like I said, points may vary a little bit, but you will see the format identical. So you'll have a short section, you'll have a fill-in-the-blank section, you'll have a problem-solving section. That's basically what's involved. Yeah?
Can the multiple choice be just one answer instead of multiple answers?
Well, okay, so if you look at those multiple answers, they're just true-false. You circle if it's true, and you don't circle it if it's false. That's all it is. So if I put little blanks and you put T or F in front of there, you would say, oh, it's a true-false question. So no, you can't do that. So, but that means you can have more than one thing true, more than one thing false. But the point is you get points for every one you get right. Right? You don't have to get them all right to get the points. You get points for every one you get right. Okay. I will do a review session tonight immediately after this class. And so the format of that will be that I take questions. I will answer your questions and hopefully answer your questions and go with that. So we're set. All right. So what I want to do today excuse me, is go through and talk some more about controlling enzymes. Because I said yesterday that controlling enzymes is a very critical function for cells to do. They have to control those enzymes, because if they don't control the enzymes, then chaos ensues. And although cells have a fairly chaotic environment, that chaos is a managed chaos. Okay? So we need to be thinking about how we can control that chaos inside. Okay. Well, I didn't show you the structure yesterday, but I did tell you that ATCase had 12 subunits. And there's actually 3 of these green on top, 3 of the green below. And these guys are 1, 2, 3, 4, 5, 6, and these are actually oriented on top of each other as well. So there's, 6 of these and 6 of these. So there's a total of 12 subunits in this overall guide. The catalytic subunits are in the center. You're not going to need to draw it, so don't worry. And the regulatory subunits are out here. As I said last time, the catalytic subunits are where the reactions are catalyzed. That's where the substrate binds. And the regulatory subunits are involved in regulating the enzyme. And that's where things like ATP and CTP bind. I noted yesterday that aspartate was also a regulator of the enzyme, but it's a substrate, so it binds actually at the catalytic subunit. The way in which these regulating molecules, these allosteric molecules, the way in which they manifest their regulation is by converting the enzyme into either the R state or the T state. Okay? And I'm going to say a little bit today, in fact, I'm going to say as the first thing, a little bit about different ways of seeing this R and T state flip. Okay? So I've talked about R state and T state, but we haven't really talked about how that can happen, so we start talking about other models. All right? Well, let's talk about the easier one of the two to understand. All right? The first model I want to talk about is called the sequential model. And it's the model I sort of described for hemoglobin. So I'm going to describe it to you in words, and you can internalize it in whatever way you want. And that is, the sequential model says that, of course, every time we have an allosteric enzyme, we've got multiple subunits. We've always got multiple subunits for an allosteric enzyme. Okay? That's given. The sequential model says that the binding of one molecule to the enzyme induces change in the other subunits. So there's a cause and effect. That is, the change in shape is caused by the binding of a molecule. So the example I've given you so far was hemoglobin. I said the hemoglobin had 4 subunits. I said binding of the first one caused the enzyme to change shape slightly, and that favored binding of a second, which favored binding of a third, which favored binding of a fourth. So the change in shape that we saw was caused by the binding of a molecule. That's the sequential model. This one, there's a sequence. This one, this one, this one, this one, right? So we see a sequential set of steps that are happening in the structural change of this enzyme. Now that's in contrast to another model. And by the way, some enzymes work sequentially, some enzymes work in a concerted fashion. So there's examples of both of these. Okay? If we think about the concerted model, and the reason I bring this up is ATCase shows some characteristics of working in a concerted fashion. Okay? What we see is this. Okay? The concerted model says that there's not a cause and effect for binding and shape change. It's a big difference. In the first case, we had a cause and effect. The binding caused the shape change, and that shape change was favored by binding it even more. We saw a snowballing effect, right? So what's causing the shape change in the concerted model? All right? The concerted model says that the flipping between R and T happens all by itself. It's not caused by the binding of a molecule. Instead, it is stabilized by the binding of a molecule. That's a very big difference. All right? So let's imagine I've got this enzyme. It's sitting here in the R state, and at a certain frequency, it'll flip on its own to the T state. So it says that flipping is back and forth all by itself on its own. Okay? It gets to the T state, and if it's in the T state and it binds to CTP, the concerted model says it will lock it in that configuration. So the binding of the molecule is a lock. It's not a cause-effect. That's the big difference between them. So the flipping is complete. It's complete from R to T. It flips all by itself. And if the right molecule binds to it, it gets locked. So if CTP binds it in the T state, it gets locked in the T state. If ATP binds it in the R state, it gets locked in the R state. Okay? Now the concerted is usually a little harder to understand, but all it is just something that's holding it in that configuration. As long as it's bound to ATP, it's going to stay in the R state. As long as it's bound to CTP, it's going to stay in the T state. Make sense? Yes? And this is allosteric. These are all allosteric. So it's also true for aspartate, if that was what you were starting to say. Aspartate does the same thing. So aspartate will lock it in the R state if aspartate binds to it. Okay? So what I'm explaining to you is how allosteric changes can happen. Either in a sequential fashion by cause-effect, binding causes the shape change, or by a locking into place. That's the concerted model. But both of those explain how allosterism occurs. Does that answer your question?
Yes.
Okay. Yeah. Okay, so if it binds to an inhibitor, does it block, does it what?
Lock in.
Okay, so it's a good question. So you guys keep coming up with my exam questions for me, so I'll try to answer your question. You're giving me exam questions, okay? If it binds to an inhibitor, what do you suppose happens? What's that? Well, but relative to these changes. That was your question, right? Well, I'll tell you the answer to the question. It depends. It depends on the nature of the inhibitor, right? We saw different kinds of inhibitors. Something that binds to the active site is going to physically block the active site. That's going to be very different. And it doesn't matter if you have R or T if you're blocking the site, right? If you bind it in a non-competitive fashion, you're not binding the active site, then we would see a different phenomenon occurring, right? So we could imagine it might lock it into that state, or it could work in either fashion in that case. It could be a concerted where it locks it in that state, or it could be a cause-effect. Either one of those is possible in a non-competitive inhibitor. But a competitive inhibitor is physically blocking it. Then there's nothing that's going to happen. Make sense? Okay. Good, good. All right. So those are the two models about how this goes. Don't worry about all these equations, and don't be worried about being able to draw this. To be honest, I look at this myself and I go, bleh. Okay? That's why I give it to you in words, and hopefully you can understand. If you don't understand that, then do come see me, and I'll talk you through it. Okay. So I hope with that I've now covered, at least at a reasonable level, how we control enzymes in one way. That's allosteric control. We're going to move on now to another type of control. And the other type of control is called covalent modification. Okay? Covalent modification. We haven't talked about that yet, so it's important that we do that. All right? Let me show you some examples. How do proteins get covalently modified? Well, you saw some examples when I talked about collagen. Remember, collagen got covalently modified by gaining some hydroxyl groups on its prolines, right? That was a covalent modification. It's making a covalent bond. Well, what you see on the screen is an example of another common covalent modification, and this common covalent modification frequently affects the enzyme's activity. Okay? Collagen is not an enzyme, so we don't worry about its activity. But enzymes that get covalently modified frequently have their activity modified. In some cases, it can be increasing the activity. In some cases, it can be decreasing the activity. Well, the most common types of covalent modification that we see, okay, there's really two types. The first one you see on the screen, that is phosphorylation or dephosphorylation, meaning putting a phosphate on or taking a phosphate off. If we go in the rightward direction, we see putting a phosphate on. If we go in the leftward direction, we see taking a phosphate off. Okay? Now you'll notice it's a serine. It turns out that there are 3 amino acids that are commonly phosphorylated or dephosphorylated. They're the 3 that have the OH groups: serine, threonine, and tyrosine. And yes, I think you should know that. Serine, threonine, and tyrosine. Now, I'll tell you why this works in a second, but I want to show you the reaction. So here's serine. There's its OH group. Notice that ATP is involved. It takes some energy to put that phosphate on there. And the phosphate comes from ATP. That's why we're left with an ADP in the end. Phosphate gets put on there, and we've got a phosphate on where there was an OH. Okay? Threonine, very similar. There's threonine. There's the side chain. There's the OH. There's the ATP. There's the phosphate getting on, etc. The last one is tyrosine. Tyrosine looks like that. There's its OH group. Phosphate gets put on, and we have a phosphorylated tyrosine. Now, why does putting a phosphate on or taking a phosphate off affect the enzyme's activity? All right. What was the charge on serine to start, or on the serine, threonine, or tyrosine to start? It was 0. What's the charge on after we put the phosphate on there? -2. Big jump, right? You can imagine if there's a negatively charged carboxyl right here, and I put a phosphate -2 charge right next to it, what's going to happen? They're going to repel, and they're going to change the shape of the protein slightly. And now we see again, changing shape of protein slightly changes the properties of the protein. In some cases, it may be favoring the protein to be more active. In some cases, it may be favoring the protein being less active. Okay? So these phosphorylation changes really, and as we will see, they're really interesting and they're really important. They really tell us some cool things about enzymes. And I'll show you some of that as we get going through stuff. Okay? Questions about that? I was so clear, or you're so thinking about the exam. All right. Let's, I want to give you one more, or 2 more terms here. Okay? 2 more terms. There are enzymes that catalyze these reactions. All right? And these enzymes that catalyze these reactions are really, we'll talk about them a lot this term. All right? Enzymes that put phosphates onto things are known as kinases. K-I-N-A-S-E-S. Kinases put phosphates onto things. So this is a kinase that puts this on. There's a specific one, and it has a specific name, but in general, it's a kinase. Okay? If I want to go in the leftwards direction, I want to take the phosphate off of something that's got a phosphate on it, I need a different enzyme. It's called a phosphatase. That's P-H-O-S-P-H-A-T-A-S-E. Phosphatase. Kinase puts on, phosphatase takes off. Okay? Now these are both important because, as I like to say, cells are the ultimate control freaks. They want to have not only an on switch, they want to have an off switch, right? Having a light switch that only goes on doesn't do you any good at night, right? When you go to bed, wasting electricity, right? You turn it on, you want to be able to turn it off. If you want to put a phosphate on, you want to be able to take the phosphate off. All right? Now what we will see happens with phosphorylation and dephosphorylation is this process right here is very important in a phenomenon that we'll talk about called signaling. Cells talk to each other. This is one of the ways in which they talk to each other. They talk to each other by controlling each other's enzymes. Okay. I'm going to skip the sodium-potassium pump because it's not really relevant to our discussion at this point, and talk about a system. Well, actually, I always contradict myself. I'll briefly mention it. Okay. Your book is kind of odd in putting it at this point because it talks about this protein that's important in our cells for pumping sodium out of cells and pumping potassium in. It's called a sodium-potassium ATPase. And the reason that they bring it up here is that during the action of this protein is, as I said, moving ions across the cell membrane, basically. During the process of action of this protein, it transiently gets a phosphate put onto it. We're going to see that transient changes, things that only last for a short period of time, that are covalent modifications, turn out to be very important for some enzymes. All right? So this is an example of a protein that gets a transient addition of a phosphate. Okay? You can see it right there, E-P. It's transient in that it only happens during part of the cycle. It goes on, and then it comes off. And it goes on, and it comes off. And it goes on, and it comes off during the cycling of this enzyme. All right? I'll say more about this particular enzyme later in the term. All right. What I want to talk about instead is this guy right here. Now, I'm just going to introduce this figure to you because we're going to talk about metabolism of this later. And all I want you to get out of this figure is the phosphorylation and dephosphorylation information. Okay, so there's a lot of information that's on here. Glycogen phosphorylase is an enzyme that we have in our liver and in our muscle cells. And it's an enzyme that breaks down glycogen. Glycogen is a polymer of glucose. It's the primary way in our body that we store glucose is as a polymer. One of the things you're going to get tired of hearing me say during the term is glucose is a poison. There's a lot of evidence that glucose is a poison. So cells have to do something with it. And rather than leave it laying around, like you wouldn't want to leave rat poison laying around your house if you had a kid, you would put it somewhere where it wasn't going to be a problem. Cells take that poisonous glucose and they make a polymer out of it. And when they make a polymer out of it, it's no longer poisonous. That polymer is glycogen. So when cells need energy, the first thing they do is they break down their glycogen. Because when they break down their glycogen, they release glucose, and that glucose can be used for energy. Okay? So this enzyme is called glycogen phosphorylase, and it's not even very well labeled on here. It's labeled phosphorylase A, phosphorylase B. This enzyme breaks down glycogen. All right? So where it says phosphorylase A and phosphorylase B, I call it glycogen phosphorylase B, glycogen phosphorylase A. What's the difference between B and A? All right? The difference between B and A is whether they have phosphate on or don't have phosphate on. So here's the enzyme. Schematically shown. It's a dimer, 2 units, phosphate, phosphate. Okay? Notice at the top, if we go to the right, we see something called phosphorylase kinase. There's that word kinase. It's putting a phosphate onto glycogen phosphorylase, and it's converting it into the A form. The A form has phosphate. And the A form is in the R state, meaning it's much more active. If I want to convert glycogen phosphorylase A into the B form, okay, all right, it can also be in the R state, and this is a little confusing, but it loses its phosphate. Notice that both of these can be in a T or an R, and both of these can have phosphate or not have phosphate. Now, what this figure, which is kind of complicated, is showing us is that the putting on or the taking off of the phosphate does not change R and T. Okay? The only thing that changes R and T, I've already told you, allosteric. Binding of small molecules changes R and T. Okay? So covalent modification is a separate mechanism from allosterism. It still affects enzyme activity. Is it active or not active? But that consideration about active or not active has something to do with what's on it, not whether it's in R or T. Okay? I know that's a little complicated. Let me give you the primary message, the take-home message here. Okay? Enzyme can be phosphorylated, dephosphorylated. Kinase puts it on, phosphatase takes it off. Okay? Bear with me there. No phosphate, phosphate. Okay. If we want to flip R and T, we have to add small molecules to it. That gives allosterism. Allosterism is separate from covalent modification. Now, we'll talk about what each of these different 4 states are later. We don't need to worry about that right now. The main point here is showing you covalent modification, and the main point here is showing you how that's different from allosterism. Allosterism converts R and T. Covalent modification converts phosphate, no phosphate. With me? I see a few nods. Okay. Now, I'll give you something you don't need to know for the exam. You can put your pens down. Yeah?
So is the covalent modification just talking about phosphate? Okay.
Yep. So is the covalent modification just talking about phosphate on or off? And the answer is yes. Okay. But binding of small molecules is going to involve allosterism. So some of these small molecules you'll see, glucose 6-phosphate, glucose, caffeine. Okay? Yeah, back in the back.
So the covalent modification does not change the shape of the
It does slightly change the shape, yes, but it's not causing the RT conversion, no. Yeah. Yeah?
Is it because it's due to the size of it, small molecules as compared to just the phosphate?
No, it's not. There's a more technical reason than that. So there are some reasonable sized molecules that will cause R and T flips, but it's, well, actually, let me back up on that. No, I would say that it is the small, they do generally have to be small, yes. Okay. But it's a more technical answer than I can give you for your question here. Okay? All right, so you can put your pens down for a second. I'll tell you a cool story about this, all right? You can still write if you want to, but you don't have to, okay? All right, so this guy turns out to be really critical, all right? Really critical, and really does some cool stuff. I'll talk about it later in the term. But one of the things this does is, how do we put phosphate on or not put phosphate on? Putting phosphate on tends to make this guy really active. It really breaks down glycogen really well. Taking phosphate off tends to make it much less active. Okay? So that's a big decision cells have to make. Do I want my glycogen phosphorylase to be really active and break down glycogen or not? Well, think about your physiological needs. Right now, other than all the brain power that you're using, I will wager that you don't need too much glucose, other than maybe to stay awake, right? You're not exercising, you're not breathing heavily, you're not doing anything that's very active. Would you want to have your glycogen phosphorylase being active right now? No, you don't want to waste your glycogen, right? You would imagine that you would have your glycogen phosphorylase in the unphosphorylated state, right? Let's imagine this room caught on fire. All right? And it's every person for themselves to get out of here, and you got to all of a sudden get up and get going quickly, right? I suppose in that state you would want to be breaking down your glycogen, right? You want those muscles to have as much energy as they can, right? Your body is releasing adrenaline. Adrenaline is known as epinephrine, and one of the ways epinephrine works is it puts phosphates onto this enzyme. I said signaling. Talking to each other. Cells are talking to each other. Adrenaline is a hormone that allows cells to talk to each other. Okay? Put this guy on. Adrenaline comes along, puts phosphate on. We start breaking down glycogen. We got a lot of glucose. A lot of glucose. That's why you hear these stories about people who can lift a car when there's danger. They're true. Your body is dumping a ton of glucose into your muscles. Okay? What causes the reversal? Insulin. Insulin. That sort of makes sense. You eat a meal, what happens to your blood glucose levels? They go high. And I just told you a second ago that glucose is a poison. So your body does something to get rid of that poison. It makes glycogen out of it. It's kind of cool. Okay? We'll talk more about that later. All right, Nathan, pick your pencils up again. All right. I've told you about one kind of covalent modification that can occur in cells, but there's another common type of modification that occurs in our bodies. And it occurs in a couple ways. All right? One of them is on the screen. So the other covalent modification that commonly occurs to control enzymes is breaking peptide bonds. Breaking peptide bonds. All right? Now, this turns out to be a really interesting and really useful regulatory scheme. Let's think about what this means. All right? My pancreas is, I love my pancreas, right? My pancreas makes my insulin. My pancreas makes my digestive enzymes so that I can digest the food that I eat. And I like to eat food, right? You guys said, no, you look too thin for that, Kevin. Okay. You don't like to eat food that much, do you? But I do. Okay. My pancreas is making those digestive enzymes, right? Now, you probably never thought about it before, and you probably never thought too much about how many people have thought a lot about their pancreas? You've thought a lot about your pancreas? Nutrition class, because they made you think about your pancreas. You didn't sit around on a sunny Sunday afternoon thinking, wow, I wonder how my pancreas is doing here, right? It doesn't happen a lot, okay? But you should. You should. And there's a reason why you should, all right? Because the pancreas is making all of those enzymes that can break things down. How come the pancreas doesn't break itself down? What's that? That's right. So the enzymes that the pancreas releases are released in an inactive form. So they don't attack the pancreas because they're perfectly capable of doing it. And if they get inadvertently activated, they will attack the pancreas and cause a very painful condition known as pancreatitis. In some cases, it's fatal. Okay? So now you see you start thinking about your pancreas more, number one. All right? Number two, you realize the importance of controlling those enzymes, because if you don't control those enzymes properly, you're going to feel some pain and you may die. You're going to lose your pancreas. Okay? So how is this done? Well, as was noted, the pancreas releases enzymes in an inactive form, and that inactive form they're called zymogens. Z-Y-M-O-G-E-N-S. Chymotrypsin that I've been talking about is a protease. It's very useful in digestion. It helps to break down proteins, but I don't want it attacking my pancreas, so my pancreas releases it in a form known as chymotrypsinogen. So when we see zymogens, one of the ways in which they're named is if they're inactive, the O-gen is put on to the end of it. So anything you see with the name with the letters O-G-E-N at the end, that will be an inactive form of an enzyme. Here's the inactive form of chymotrypsinogen. Activating chymotrypsinogen requires action, okay, in two steps. The first step involves breaking a bond right there, okay? And all it's doing is just breaking that bond, making an opening there. This creates an enzyme that is partly active. It says active enzyme. It's only partly active. It's called pi chymotrypsin. Pi chymotrypsin. Okay? Pi chymotrypsin is able to degrade other pi chymotrypsins and make them fully active. And making them fully active involves removing a few amino acids, not very many, you see. And we see these blue guys holding everything together. What do you suppose those blue guys are? What's that? Disulfide bonds. Excellent. Okay. So we got cysteine-cysteine disulfide bonds. That's holding everything together. Notice that they formed way up here. What did that tell you? The folding of the original protein was important. It had to fold first before this was even possible. So it folds. The right links get made between the cysteines. It folds. And that's going to be important because if it doesn't fold properly and these guys don't get together, then by the time we do these cuts, all these pieces are going to fall apart. So the folding happens, then the cleavage happens, and leaves us with a final chymotrypsin that is active. Okay? Now, you might say, oh, well, this takes the problem one step away. Trypsin, won't it attack the pancreas? No, trypsin is released in an inactive form called trypsinogen. All right? So it turns out that there's a hierarchy of these active versus inactive, and they tend to work in what are called cascades. One activates the next, activates the next, activates the next, etc. And the body has a good way of protecting itself so that only further in the digestive system, further along down the digestive system, do these activations occur. Now you're probably thinking, well, okay, Kevin, I understand it's not going to attack the pancreas, but isn't it going to attack my intestines? And the answer is, yes, it will, to some extent. But some of the most rapidly proliferating tissue in your body are tissues in your intestines. Okay? They're designed to slough off, make new cells, etc. Okay? They're really designed very well for the function that they provide. So you're willing to give up a lot of intestinal cells to be able to digest all the other stuff that you eat. It's when you have trouble replacing those digestive cells that you develop things like ulcers and pain. And there's some biochemical reasons for that. We'll talk about those later. Okay, questions about this? So we've gone from an inactive enzyme to an active enzyme. All right. Okay, now let's spend just a little bit of time talking about how chymotrypsin works. All right. How does chymotrypsin work? How does a protease work? We talk about chymotrypsin, it's probably the most studied of all the different proteases. How does it work? All right. Well, it turns out this enzyme is not what anybody expected when they first started studying it. It wasn't what they expected when they first started studying it. Okay? If you recall, I told you biochemists were lazy, and I told you that we used things that gave us color indicators to tell us how fast chymotrypsin was working. Just to remind you, that mechanism was right here. Okay? We cheated. We made something that looked like a peptide bond for it. Chymotrypsin acted on it, and it created something that gave a color. Okay? This color over here. This guy's colored. So we can follow the reaction by seeing how rapidly the color appears. When people started studying chymotrypsin, something surprising occurred to them. The surprising thing was right here. They discovered that, now notice what we're plotting here. We're plotting the rate of the reaction versus time. That's not concentration of substrate, but against time. Okay? What they saw was the reaction starts off fine and dandy in a nice linear fashion, and then it sort of bends over. When they saw this, they realized that the reaction was actually occurring in two steps. A fast step and a slow step. So the reaction that chymotrypsin and most proteases catalyze occur in a fast step and a slow step. That fast step and slow step will be the subject of what I will finish with today. So I'm going to tell you a bit about mechanism. This is the only time in the term I'm going to talk about mechanism, but we do need to spend some time talking about mechanism. All right? All right, this looks kind of hairy. I'm going to try to keep it basic and simple for you. Okay? First of all, let's think about what chymotrypsin does. It breaks peptide bonds. That's what it does. It's a digestive enzyme. It breaks peptide bonds, meaning it's breaking proteins down into big pieces into smaller pieces. That's what it's doing. It does that by breaking peptide bonds. So the mechanism that we're talking about here is how it breaks peptide bonds. All right. Now, I could teach almost a week on this. I'm not going to do that. All right. So I want to give you the highlights of what is involved in this catalytic mechanism. All right. What you see on the screen is up close and personal the active site of chymotrypsin. Chymotrypsin is an enzyme that we call a serine protease. Serine protease. First of all, I told you a protease is an enzyme that breaks proteins down. So chymotrypsin is a protease. But more importantly, it's a serine protease, and that tells us that a serine is involved in the catalytic mechanism. Serine is involved in the catalytic mechanism. There are many serine proteases, and they all have a virtually identical mechanism to what I'm going to show you. Okay? Now, if you understand something about the mechanism by which this enzyme works, you can understand the mechanism by which almost any enzyme works. There's some very general principles that you'll be able to use elsewhere here. Okay. Active site of the enzyme. That's the place where the reaction is catalyzed, right? That's the place where I've got to have that bond I'm going to break, right? So the enzyme has to grab a hold of another protein and hold on to it in its active site. Right? All right. How does it do that? Well, the way enzymes find their substrates is by random bumping into each other. Okay? Bumping into each other. You notice when I talked about trypsin that I said it worked by cutting next to lysines and arginines. That's a recognition for that enzyme to say, oh, you got the right thing. Hold on to it. Chymotrypsin does that as well. It doesn't use lysines and arginines. It uses some other amino acids. But once it recognizes an amino acid, it says, oh, I've got something I can cut. Right? If I bind the right amino acid in this protein, I've got something I can cut. All right. When that happens, okay, there's a series of steps. Binding of the proper substrate causes, and you've already learned what happens when the enzyme binds a proper substrate. What did we call it yesterday? Koshland gave it a name. The shape changed because the substrate bound to it. What was it? The induced fit, right? The induced fit said that not only does the enzyme change the substrate, but the substrate transiently changes the enzyme, right? It's going to change the shape of the enzyme very slightly, right? Now we're going to see how that's going to happen. All right? The proper substrate has bound in the active site of the enzyme. The enzyme is now going to undergo a very slight shape change, and that shape change is going to enable the reaction to occur. That's what's happening. I just lasered myself. That's what's happening here. Okay? I want to take you through that. In the active site of the enzyme, there are 3 amino acids that play very important roles in the catalytic action of the enzyme. This is part of the enzyme now. 3 amino acids in the active site of the enzyme play very important roles. When there's no substrate bound, these 3 amino acids, they're called serine, histidine, and aspartic acid. They're just laying here in the active site doing nothing. The proper substrate binds, and now I told you the enzyme changed shape very slightly. When that slight shape change happens, these guys get closer to each other. Serine, histidine, and aspartic acid get closer to each other. That's the key to how this enzyme works. They only get close to each other when the proper substrate binds. Okay. Now, here's serine, here's histidine. Over here, it's not shown, is an aspartic acid. Okay? Aspartic acid has a side chain that is negatively charged. It's a COO minus. You learned that, I hope, from your amino acid structures, right? At physiological pH, it's negatively charged, COO minus. If I put a COO minus right next to this histidine, COO minus means it's got extra electrons, right? Electrons are negatively charged, right? What happens if I put a bunch of negative charge right next to a ring that's full of electrons? Well, those electrons are going to move away from the negative charge. They're going to exist over here instead of existing over here. They've just shifted slightly. So the electrons in the ring have shifted. They've moved in the direction of serine. That slight shape change has caused the electrons in histidine to move over closer to serine. Well, serine turns out to be really useful because serine has an OH out there. And that H looks really attractive when there's a lot of electrons here. That H is positively charged if I take it away. And that can neutralize the negative charge that's in that ring. And that's exactly what happens. This histidine, that's what's being shown here, takes away the proton off of serine. It takes it away. Well, needless to say, that causes a little angst on serine. Serine's not too happy with this because serine has just lost a positive charge, and if I have something that has a 0 charge and it loses a positive charge, what's its charge? Negative, right? What I've just described to you is the first step in the process of this breaking this peptide bond. We haven't talked about the peptide bond yet. But we're getting everything ready to break the peptide bond. Okay? We just created something called an alkoxide ion. A-L-K-O-X-I-D-E. Alkoxide. A-L-K-O-X-I-D-E. Alkoxide ion is that negatively charged oxygen that's left behind. Everybody understand how the negatively charged oxygen got made? Shifts, proton pulled, negative charge left behind. All right. That negatively charged oxygen is not content. As I said, it had some angst. It attacks the peptide bond. Okay? It attacks the peptide bond. Now, that attack of the peptide bond causes the peptide bond to break. All right? Causes the peptide bond to break. So where we had a long polypeptide chain and we break it in the middle, which is what we've just done, where we had one piece, we now have two pieces, right? Everybody understand? We've taken something that's a one piece, broken it in the middle, two pieces. Well, one of those pieces is completely free and it floats away. It's completely free and it floats away. The other piece turns out to be covalently linked to the oxygen in that serine. It's covalently linked to it, and that's what you see actually right here. It's stuck on it. Now, if you remember, I said this reaction occurred in two steps, a fast and a slow. You've just seen the fast step. The fast step involved the binding, the alkoxide ion, the breaking it, and the release of that first piece. Bang, 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 bang. It happens very quickly. Now we're ready for the slow step. The slow step means we got to get this thing off of the serine so we can get back to where we started. Okay? That slow step takes a few steps, and I'm not going to take you through the mechanism of that. It simply involves water coming in and breaking the bond between that other piece and the serine. Water has to diffuse into the active site, and that takes a while. Now, when I say a while, we're talking on the order of milliseconds to seconds, not a long time. We're not talking hours, for example. But this first part took place in milliseconds. Bang, happened very quickly. Second part happens more slowly. After water comes in and breaks that bond, we're back where we started. And the enzyme can go through a complete new cycle. Now, what I've just shown you is that enzymes can transiently be bound to something. In this case, it was transiently bound to one of the polypeptide chains. Question?
Just wondering,
Yeah. So how does the carbon have 5 things attached to it? You think they made a mistake there, right? Okay. This is a transient intermediate, just showing you all the possible electrons that are out there. This turns out not to be a stable intermediate. And since I'm not going into the mechanism, I won't tell you how it's resolved. But that unstable intermediate has to be protected inside of here, because if it's not protected properly, it will react with the enzyme and inactivate the enzyme. So good eyes, very good eyes that you saw the 5 bonds. Yes, ma'am.
So in these pictures, is everything in the brown inside? Is that perfect, or is that on the outside?
Good question. So all these are occurring. So whenever we look at an enzyme and we see its catalysis occurring, the active site is almost always on the inside of the protein. So yes, it is on the inside of the protein.
That's all reactions within itself.
These are all reactions that are occurring inside of the protein. That's correct. But it's not with itself. It's to this other peptide that it's bound to. Remember, it's cutting something else that it's bound to. Right? So this thing right here is the external protein that it is bound to in its active site. That's the thing it's cutting in half. You like mechanism? I don't either. Okay. Questions? Yeah.
How much of that will we actually actively produce, or can you just basically give a general outline?
Well, my rule on this will be the same as any other rule. That is, whatever I talk about in class is fair game.
But do we need to be able to draw the steps of that?
I'm not going to ask, it's not an art class, so I'm not going to ask you, because if I ask people to draw art, then art is the hardest thing in the world to grade. Okay? But I think that you should be able to describe to me what I've described to you. I think you should know what's there, yeah. Okay? At the level I've told you about it. Now, your book will go in, the book will go into a little bit more depth with it, but at the level I've talked to you about it here, that's I think, reasonable. Yeah?
So the initial shift of the electrons, is that caused by the outside protein bonding?
Okay, so our question is does the initial shift of electrons happen as a result of the binding of the external protein? And the answer is yes, absolutely. It's that binding, remember, that's causing that small shape change, and that small shape change is what's pushing that aspartic acid next to that histidine. So we see a sequential series of steps that's happening in this catalytic action. Now, I want to use one more word with you, and then I'll finish for the day. And that one more word is this alkoxide ion, this reactive oxygen that's here. It's electron rich. It normally has a proton there. We don't normally see protons come off of serine. I told you it's very reactive, and the reason it's very reactive is it's what we call a nucleophile. It's seeking a nucleus. The nucleus it's seeking turns out to be right there in the active site. It's that carbon of the peptide bond. Okay? That's what's happening. Yes?
So is the substrate still on at the end of this?
No. By the time it's done, water has come in, and it releases that bond. That's the slow step. I said I didn't show you the mechanism, but water breaks the bond between the serine and that remaining peptide, and the remaining peptide gets released. So the first piece comes off very quickly. The second piece comes off very slowly. But they both do come off. Make sense?
It probably happens in different parts of the body. It binds, it moves, all this happens, and then it kicks off.
It's all happening in one place. Remember, this is in an enzyme now, not in a structure in the body. So in the enzyme, it's going through that very first step very rapidly, cutting it in half, throwing away one of the pieces. The other piece it's covalently bound to, and then within a second or two, it's going to go through the rest of this process and release that. Does that make sense? Okay.
What's the overall objective of it?
The overall objective is cutting a protein. So it's cutting a protein in half. I start out with one piece. The enzyme acts on it, and I've got 2 pieces. That's important in digestion because I'm taking a big molecule and breaking it into smaller pieces. Okay? Yeah, back here. Say it again. Say it again. Yep. So when I cleave the peptide bond, what I'm doing is I'm regenerating a carboxyl group, just like I had originally, so it's not going to be reactive. And then I regenerate the amine group, and it's not going to be reactive. So the answer is no. It's not reactive after it's been broken. Okay? Yeah?
So the alkoxide ion, I mean, that's
Okay, so his question is, what happens if the alkoxide ion binds to something other than the protein? This is a really cool thing that I want you to understand. I'm glad you asked the question. Okay? The alkoxide ion will only form if it is bound to a protein. Because remember, it's the proper binding that causes that shift that makes the alkoxide ion happen. And that only happens when the proper substrate binds into the active site. So you will not have an alkoxide ion unless you have the proper substrate that is bound. That proper substrate is a protein. Does that answer your question? And some of you had a question back here. You had a question? Yeah.
I was wondering, going back at the graph, you had that boost area.
Yeah.
Does the top of that boost area signify when all the enzyme is, like, say, tied up
Yep.
with the slow step?
Okay. So let's go back to the graph that you're asking about. So right there, right? Okay. I want you to keep in mind that when we do a study like this, we have not one enzyme molecule, but we have 100,000 enzyme molecules. Right? So what we see in this phase is we see those that are quickly making it through, releasing that first step, and then with varying efficiencies, they will be releasing the other one.
So I guess my question is, when it starts to go slower,
Yep.
does that mean that the enzymes are all saturated with the slow step?
So it means that the slow step is determining the overall rate. Because until that slow step gets resolved, it can't go back and do another fast step. And that fast step happens, and it's got to wait again. So the limiting thing is the slow step. And that's what this bend really tells us. Down here, there's no slow step. Up here, there's slow step and fast step. Right? Is that a question?
Yeah. So it cuts one peptide bond and then releases, and then it just keeps doing it until the molecules are all broken down.
So it's going to. So her question is basically about the mechanism by which this breaks down. So when it breaks it down, does it just keep moving along? And the answer is no, it doesn't. Okay. So remember what it's going to do is it throws the first one out. So it releases a smaller peptide, and then it throws the second one out,
Right.
and then it goes and finds a new one to bind.
So
So
those two pieces don't ever get worked on.
those two pieces may get worked on by other enzymes, but not that particular enzyme. That's right. Okay. Other questions? I had a silly song to sing. We can sing it if you want, if you'd rather we do the review session. What's that? Review? Okay, that's good. Let's do that. Let me turn the camera off and turn the camera back on, then we'll start the review session.